I'd like to thank you, the committee of the International Webinar on Cognitive and Behavioral Neuroscience, for opportunity to share with neuroscientists our result concerning the anti-inflammatory effects of omega-3 PUFAs in astrocytes. We are aware the fundamental function of astrocytes in all aspects of the CNS functionality, from maintaining an overall brain homeostasis, neuronal support, protection against oxidative stress and exocytotoxicity, up to maintaining cognition through a human life by adaptive neuronal plasticity and regeneration after injury. However, astrocytes contribute also to worsen outcomes after various CNS insults, among other through failure or attenuation of the glutamate-glutamine cycle, increased destructive inflammation after the prolonged activation by active microglia, or intensified infection due to failure of the blood-brain barrier, where astrocytes play a crucial role. A prominent negative role the astrocytes play in neuroinflammation across neurodegeneration disorders and brain aging in humans. The resident macrophages, microglia, depend on the inflammatory context, upregulate numerous membrane receptors as TLRs, CD33, CD36, complement receptors, and RAGE, enhancing their sensitivity. Even more importantly, they flood vicinity of the brain parenchyma with pro-inflammatory mediators such as IL-1, IL-6, TNF-alpha, MCP-1, interferon gamma. A prolonged or intense response of macroglia triggers transformation of resting astrocytes into the neuroinflammatory phenotype called I1 astrocytes. This type of astrocytes have changed morphological features such as potent ramification, upregulation of GFAP, and enhanced production of pro-inflammatory cytokines and mediators that enhance neuroinflammation, creating a vicious cycle. Taking into account the favorable function of astrocytes playing under physiological condition and importance of efficacy restoration of senescent astrocyte associated with increased oxidative stress, enhanced reactivity on various insults, we investigated the effect of DHA alone or DHA and EPA on astrocytes, bearing in mind they widely report anti-inflammatory effects in vitro study, in animal model study, and clinical trial. A potent anti-inflammatory effects were reported by Lu and co-workers, which showed that DAH reduced expression of pro-inflammatory cytokines and enzyme induced by interferon gamma in microglia. Later, the group of Petit discovered that next to reduction of pro-inflammatory response in microglia by DAH, also there was increased the total glutathione levels and antioxidative capacity of these cells. In 2012, Labrousse described that increased content of omega-3 PUFAs in the brain after dietary supplementation prevented cytokines expression and restored special memory deficit in the hippocampus of aged mice. The other study by Harvey and co-workers revealed that in DHA-treated rats, decreased nuclear translocation of NF-KB and reduced neuronal degeneration result from traumatic brain injury. Su and al. In the clinical study reported that supplementation with omega-3 PUFAs in patients with hepatitis C virus receiving interferon alpha therapy significantly delayed the onset of interferon-induced depression. Numerous papers indexed to the clinical trial omega-3 and Alzheimer's disease demonstrated a positive outcomes on cognition as well anti-inflammatory effects. For example, Vedin and co-workers reported that omega-3 PUFAS supplementation increased the level of PUFAS in plasma that was negatively correlated with secretion of inflammatory cytokines from the blood mononuclear cells of these patients. In our department, we working on the primary rat astrocytes and astrocytes derived from human-induced stem cells incubated with omega-3 fatty acids. First, we establish cytotoxicity of DHA and EPA in the range of 10 to 100 micromoles during 24-hour incubation. Toxicity of DHA was slightly higher than EPA. Then we choose 30 micromoles as a safe concentration for both PUFAs. In addition, we checked time-dependent toxicity of DHA and EPA in the concentration of 30 micromoles up to 72 hours and confirm the incubation for 24 hours with this concentration is safe for the cell. Next, we determined the fatty acid composition of plasma membrane by GCMs after incubation of astrocytes with DHA and EPA in equal concentration. As you can see on the graph, the percentage of saturated fatty acids didn't change visible after incubation with omega-3 fatty acids. However, DHA doubled in contents in membrane, decreasing omega-3 PUFAs and monounsaturated fatty acids. Incubation of astrocytes with the same concentration of EPA increased both omega-3 fatty acids, DHA by 2% and EPA by 0.6%. At first, we reported a prominent antioxidative effect of both omega-3 PUFAs in H2O2 treated astrocytes, which manifested in lowering of ROS generation, upregulation of glutathione-dependent enzymes, antioxidative enzymes, and associated with increased expression and activity of NRF2. In the next study, we stimulated DHA-enriched astrocytes with interleukin-1 beta and determined activation of NF-KB and AP1 transcription factors, the expression of pro-oxidative enzyme COX-2 and INOS, and secretion of TNF-alpha and interleukin-6. Immunofluorescence and Western blot assays show inhibition of NF-kappa translocation to the nucleus, which is seen in the bottom left merge microscope photo. And also this reduction or retention of NF-kappa in cytoplasm was associated with reduced phosphorylated IkBa inhibitor of NF-KB. This inhibition was DHA-dose dependent. Along with inhibition of NF-KB activation, we've detected reduction of the c-Fos and c-Jun to the DNA sequences named TRE element. Anti-inflammatory effects of DHA-enriched astrocytes was manifested by reduction the expression of two important pro-inflammatory enzyme COX-2 and INOS, also dose-dependent inhibition of release TNF-alpha and interleukin-6. In the subsequent study, we used the similar protocol for astrocyte activation, but we incubated cell separately with DHA and EPA. We determined then proteasomal chymotrypsin-like activity and expression of inducible proteasome subunits beta-1, beta-2, and beta-5 to check if omega-3 PUFAs inhibit assembly of immunoproteasome expressed in reactive astrocytes. The immunoproteasome is transformed constitutive proteasome in which 3 the 20 S-core subunits B1, 2, and 5 are replaced by their inducible counterparts. Usually, this subunit replacement happened primarily under stimulation with interferon and TNF-alpha released by activated microglia. To be fully mature, immunoproteasome have to combine with the activators at its two ends. The activators can be PA700 or PA28. Then, in this case, assembly of core with the regulatory subunits arise variants of immunoproteasomes. Immunoproteasomes have enhanced chymotrypsin-like activity because subunit 1, inducible subunit 1, instead of caspase-like activity, possess chymotrypsin-like activity, and other inducible subunits have similar activity to their constitutive counterparts. IB2 has trypsin-like activity and B5 chymotrypsin-like activity. Immunoproteasomes are formed in reactive microglia and A1 phenotype of astrocytes being in sustained pro-inflammatory and pro-oxidative milieu. Immunoproteasome formation is needed primarily for generation of peptides with hydrophobic C terminus that can be processed to fit in the groove of MHC class I molecules necessary for expand of adaptive immune response. The upregulation of immunoproteasome were reported over the course of Alzheimer's and Huntington diseases and in neurons of aging hippocampus. Oppositely, the inhibition of immunoproteasome activity was associated with decreased interferon gamma-dependent expression of pro-inflammatory cytokines in microglia, attenuation of disease progression in neural models of Alzheimer's disease, and experimental encephalomyelitis. The incubation of resting astrocytes and stimulated with interleukin-1 didn't change the expression of constitutive and inducible subunit beta 2. However, those fatty acids inhibited inducible beta 1 and beta 5 subunits. This is more evident when we see the ratio of beta 1 constitutive to inducible subunits and the ratio of constitutive beta 5 to inducible beta 5 subunits. Since inducible beta 1 and beta 2 genes possess the cat box, we measure activity of two CEBP members of the cat enhancer binding protein family. These transcription factors are activated by numerous pro-inflammatory factors and cytokines and regulate the expression of genes with cat box important in sustained inflammation, among other the enzyme and cytokines assayed in our previous study means COX-2, INOS, interleukin-6, and TNF-alpha. As expected, pre-incubation of astrocytes with both omega-3 fatty acids reduced activity of CBP alpha and beta. IkBa seems to be more effective in inhibition of bounding CBP transcription proteins to DNA because was active already from the concentration of 10 micromoles. In the last step of our research, we investigated whether a downregulation of immunoproteasomes may prevent also degradation of connexin 43 because variable harmful conditions rise in tissues such as ischemia and induced inflammation led to CX43 degradation. Astrocytes are extensively coupled by the gap junction, essential for creation the astrocytes network for intercellular communication through calcium wave or hemichannels, allowing exchanges between the cytoplasm and extracellular fluid. Hemichannels were shown to be permeable from gliotransmitters like glutamine, glutamate, ATP, and glycerin, well-known modulators of neuronal excitability and synaptic plasticity. The major constituent of the gap junction in astro is the connexin 43, which at present was confirmed by immunohistochemical staining and specific antibodies. Immunofluorescence of CX43 in omega-3 fatty acid enriched astrocytes was slightly increased, but in interleukin-1 treated astrocytes, incubation with omega-3 fatty acid significantly prevented CX43 loss and degradation, meaning that the gap junction disassemble walls were protected. In summary, our results show that enrichment of astrocytes with omega-3 fatty acids has powerful anti-inflammatory effect by decreasing activation of NF-KB, the APA1 transcription factor, and two CEBPs proteins, downregulation of expression pro-inflammatory COX-2 INOS secretion of cytokines TNF-alpha and interleukin-6, also downregulation, expression, and activity of inducible proteasome. In addition, omega-3 fatty acids prevented loss of CX43 protecting the astrocytes network connection. Our study was published in pharmacological reports in BMC Neuroscience and MDPY International Journal of Molecular Scientists. I'd like to say thank you for participating in the webinar and present our results, and add that our research was supported by grant from the National Center for Research and Development in Poland and in frame of the Poland-Taiwanese cooperation, also by the Medical University of Łódź. Thank you so much for your attention.